Hey guys, it's Stratton. I wanted to talk today about Hardy-Weinberg equilibrium, which is an indicator of evolutionary change. It's how we measure that evolution is taking place in a population. So before we begin, I want to point out you're going to need a calculator for this. So go ahead and grab one. Before we start, I do want to go back and review the RP from last time for topic 7.4. So for each of the following examples, identify the force of evolution at work and predict the consequences for the gene pool. So number one is a small group of people shipwrecked on Gilligan's Island. Well, as you can see from the image there on the right, those people are all white. You've got a redhead, and if you didn't watch the show, you may not realize one of those people on there is actually a professor. He's supposed to be brilliant, can make a telephone out of a coconut and that sort of thing. But what would you wind up with in the future on that island? So first off, that's going to be the founder effect. And in the future, you'd expect to have a lot of white people on this tropical island, and you'd expect to have a lot of redheads on this island as well. Okay, the second example is Safari Park brought in 2 new male zebras from a zoo in Texas. That's gene flow, and that's going to increase the diversity of this population. Number 3, the northern elephant seal that was hunted to the point of extinction. So that's a bottleneck, so bottleneck effect, and it's going to reduce the genetic diversity of that population. In fact, it's going to make a population much more susceptible to diseases and things like that. Okay, number 4, female peafowl always choose to mate with the male with the largest, most colorful tail. That is called assortative mating or non-random mating, and the result is it's going to decrease the diversity of that population, and it's going to lead to an increase in whatever trait the male has, obviously, sexual selection. And number 5, plants that flower and produce seeds earlier have a greater reproductive success in a warming world. That's natural selection, and that's going to lead to a shift in the population in one direction, in this case, towards earlier flowering. Okay, for the 3 different images shown here on your screen, the one on the left with the birds, that's going to be stabilizing selection, where they only have 3 eggs, versus, if they lay just 1, that's not good fitness. If they lay a lot more than that, then they can't really care for all those offspring. The middle one is diversifying, where you're going to have 2 different morphs of jaguar. The result is that anything that's in between is not going to camouflage well. You've got the spots, that's going to camouflage really well amongst the foliage. You've got the dark, the melanistic phenotype, that's going to camouflage well in the shadows. Something in between is really not at home in either one of those locations. And then the moth over on the right, that's an example of directional selection. And that's going to, you've got an orchid that has a really long nectar tube. And so it's selected for moths with longer tongues that are able to drink from that. In fact, Darwin never saw this moth, but he did see this orchid, and he predicted that this moth would exist. It wasn't until years later that somebody actually found the moth with the tongue to be able to drink the nectar from this flower. Okay, moving on today, talking about Hardy-Weinberg. First, I want to go over a little bit of background. So the first thing is alleles and genes. So alleles are different forms of a gene, and in this case, let's say that a gene is like the word car, right? Car is a generic term. It refers to anything that has a certain function, right? So this gene has a function. The function is eye color. Allele is like the different version of that. So example, Chevrolet or Ford or Toyota or whatever, right? In this case, so we have a big B and a little b allele that are both the eye color gene. Now again, I want to point out I am simplifying. In real life, your eye color is controlled by like 8 different genes, and they all kind of blend together. It's not just, oh, you've got big B is brown, like we made it out to be in freshman biology. Okay, so at this point, you should be able to explain in your own words what the difference is between a gene and an allele. So every organism has 2 copies of each allele that they get from their parents. And of course, a little bit of review, obviously, homozygous dominant, heterozygous, homozygous recessive, you should know those terms by now. But let's imagine for a minute that we're talking about a population of a school. You have 2,000 students. So because you've got 2 alleles for each gene, you have 4,000 alleles at that school. Now, if I were to go around that school and look deep into everybody's eyes, or actually do a DNA test and look and see what alleles they have, then I would be able to figure out the gene pool of the school. So let's say I did that. I walk around the school, I take a DNA sample from everybody, and I figure out, are they big B, big B, big B, little b? Obviously, little b is easy to see because I can just look at their eye color. But if I found I had 3,200 big B alleles out of my 4,000, then I would say that my gene frequency, my allele frequency is 0.8 or 80%. So 80% of the alleles that I found when I went around the school would be big B. And then, I mean, obviously the other 20% are little b. So those numbers are going to be important. In a gene pool, like these mice here, this is the rock pocket mouse. So if we've got 2 alleles for each mouse, then that little green box there is indicating one mouse. That mouse is heterozygous. It's got a black allele and a brown allele. Okay. Obviously, in this case, black is dominant because you can see that the heterozygous are black. Okay, so if there's 25 mice, that's going to give me 50 alleles. So if I find there are 20 out of those 50 alleles are big B, then 40% or 0.4 would be the allele frequency for big B, and then the other 60% would be for little b. By the way, just to point this out, just because something's dominant doesn't mean it's the most common allele. In the case of blood type, for example, type O, which is recessive, is the most common blood type on Earth. There is another way for us to look at this too. So imagine that you're just told that you've got 4 mice that are big B, 12 that are big B, little b, and 9 that are little b. What I could do here is I can look at this group here, and really, because there are 4 big B's, that means that there are 8 big B's right here amongst those individuals. With my 12 big B, little b's, that means there are 12 big B's and there are 12 little b's. And of course, then over here, with 9 big little b's, that's going to give me 18 little b's. So what I could do then is I can say, if I have 18 plus 12, that's going to give me 30 little b's. And over here, my 8 and 12 for big B is going to give me 20. So it's the same numbers, right? If you don't actually have a picture of the alleles like we've got here with these little spheres, then you can just look at the individuals and figure out how many. So 20 out of 50, 30 out of 50, and those are exactly the numbers that I've got over here. So moving on, the key with Hardy-Weinberg is that it gives us a chance to figure out if evolution is taking place, because if evolution takes place, then allele frequencies change. The population, the gene pool changes. So in this case, when the frequency of the dominant recessive alleles don't change, that's called Hardy-Weinberg equilibrium. Now, that's telling us that there is no evolution taking place. So if the frequencies ever change, evolution is happening. Now, to be realistic about this whole thing, evolution is always taking place. So in this case, if we said that our big B brown allele and our little b blue allele are, again, 0.8 and 0.6, or excuse me, 0.8 and 0.2, if those numbers ever change, it means evolution is taking place. So if the frequency of the recessive allele is 20%, what would you expect the frequency to be in 100 years if it's in equilibrium? Well, obviously, things aren't going to change if it's in equilibrium. Now, has the population of our school evolved over time? Do you think that the blue-eyed and brown-eyed alleles are the same as they were 50 years ago? Probably not. So there are 5 conditions that have to be met in order for Hardy-Weinberg equilibrium to take place. Number 1, the population has to be big. The goal there is to avoid genetic drift, right? So example, if you've got gene flow, that would be migration. That you can't have any gene flow. No mutations. You have to have random mating, right? You can't have people choosing their mate based off a characteristic, and there can't be any natural selection. So, and the genetic drift example, that'd be like the bottleneck effect or the founder effect. You're trying to avoid that kind of thing going on. But really what these are the 5 forces of evolution. So when the 5 forces of evolution are present, the allele frequencies will change. So, and because these things are not realistic, evolution is always happening. Okay, you're always going to be given this set of formulas on the AP formula sheet. So we're going to refer to the frequency of the dominant allele as P and the frequency of the recessive allele as Q. That's just so we don't keep having to change the letters around. What you're looking at here then with these formulas, P squared, what that's referring to, so P is the dominant, Q is the recessive. But if something is homozygous dominant, then what that means is if it's, say big B, what are the odds of getting a big B? Well, that's going to be P. What are the odds of getting a second big B? You're going to multiply those two odds together. So this is P squared. Okay, it's the same thing with the recessive allele over here, Q. Little b and little b, the odds of getting little b would be Q. Oops. And the odds of getting another little b would be Q. Multiply those together and that's Q squared. And then finally for the middle one here, there are two different ways of getting big B, little b, right? You have big B, little b, and you've also got little b, big B. When you have two different ways of getting something, you're going to add their odds together. So this is PQ, this is QP. Really what you're looking at then is 2PQ. So each one of these, this is big B, big B, big B. This one here is big B, little b, and this one here is little b, little b. That'll make more sense when we start doing some examples, I think. So P and Q are allele frequencies. So that's just talking about the frequency of the big B or the little b. Genotype frequencies, like I just went over a second ago, P squared, 2PQ, and Q squared. And phenotype frequencies. So this is where you have to realize that when we're talking about a regular dominant recessive relationship, then you've got two different ways of having brown eyes. You can be big B, and you can be big B, little b. And so you have to add those two things together. There's only one way you can be blue-eyed, and that's to be little b. That's going to be important in a second here too. So in our hypothetical class, if you've got 80% is the frequency of big B, that means P is 0.8 and Q is 0.2. Those are always going to add up to 1 because 100% of our population is represented here. So this is really 100% is what the 1 is representing in that case. Okay. So what percentage of our class would be homozygous dominant, homozygous recessive, and heterozygous? Take a second, pause the video, and go ahead and figure those out for yourself. Okay, so you paused the video and you went ahead and calculated these, right? And you calculated that this is going to be 0.8 squared, or 0.64. The recessive is going to be 0.2 squared, so that's going to be 0.04. Make sure you get the 0 in there. We're going to talk about some possible errors in a second here. Whenever you multiply a decimal, it gets smaller. And finally, 2PQ, that's going to be 2 times 0.8 times 0.2. That's going to be 0.32. Now, if we check, all of these should add up to 100%. And they do. So this is 1.0. Okay. So some common errors. Since we can't really tell the difference between big B, and big B, little b, the only phenotype we really care about is little b. This is the blue eyes, right? We only really care about recessive. So when you're given a problem, you automatically key in on the recessive phenotype. Learn how to use the square root key on your calculator, because remember, if you take a square root of a decimal, then that number is going to be larger. If you multiply two decimals, that number is going to get smaller because you're multiplying by something that's less than 1. And when you're reading a Harvey-Weinberg problem, you always look for the word allele. If you see the word allele, they're giving you P or Q. That's an easy problem. Those are simple problems to solve. But if you don't see the word allele, they're giving you a genotype or phenotype. And you're going to have to figure out what you're going to have to work with there and use a square root key. So let's go ahead and do a little bit of practice together here. So in a population of guppies, spots are caused by a dominant allele, big A. If the frequency of that is 0.6, what's the frequency of the recessive allele? Well, I know that P plus Q is going to be equal to, plus Q is going to be equal to 1. And this is P, 0.6. So plus what is equal to 1? Well, of course, Q is going to be equal to 0.4. What percentage of guppies will have no spots? Well, it said spots are dominant, so no spots would be Q squared. So that's going to be 0.4 times 0.4 or 0.16. For spots, there's two ways I can figure this out. One, I know that all the ones that don't have spots, the 16%, all the rest do. So I can just say, oh, it's going to be 1 minus 0.16. That's going to be 0.84. If I wanted to take the long way about this, I could do p squared plus 2pq. And that would then give me, well, p squared is 0.36, and 2pq is 2 times 0.4, 0.6 times 0.4. So this is going to be 0.48. So if I add those two together, I get 0.84, 84%. Now, if there are 400 guppies, how many would have no spots? Well, that's going to be 400 times my gene frequency of 0.16. So that's going to be equal to 64 guppies. And now I could just subtract from 400, but just to make sure, 400 times 0.84 is going to be equal to 336. So those two together add up to 400. Okay, if you need to pause and go back and review that, do that now. Okay, an additional problem. Humans, 10% of the population is left-handed, a recessive trait. What are the frequencies of the dominant recessive alleles? So if I'm given this, I don't see the word allele anywhere in there. So I'm not given P or Q. What I'm given then is left-handed is recessive. This is the phenotype, so this is Q squared is 0.10. So if I take the square root of 0.10, that's going to give me 0.32, and that's Q. Okay, now the other way to figure out then P, I could go through this whole rigmarole, or I can just say, hey, look, these two have to add up to 1, so it's going to be 0.68 is P. Okay, what percentage of people are heterozygous for this trait? Heterozygous is 2pq, so that's going to be 2 times 0.68 times 0.32, and that's going to be equal to 0.44, 44%, and I am rounding here too. Finally, how many people are going to be left-handed if my population is 2,000? So I've got 2,000 times, and I already said 10% of them are left-handed, so that's going to be 200. How many are going to be right-handed? Again, two ways I can do this. I can just subtract from 2,000, and it'd be 2,000 minus 200 is 1,800, or I can actually do my p squared plus my 2pq. So that's going to be, I said p was 0.68, so 0.68 squared plus 2 times 0.68 times 0.32. And if I add this all together, so this is going to be, let's see, 0.68 is squared. Let's see, I don't have that one, I don't get calculated. So I think it's 0.46 plus, then I got 2, oops, sorry about that. 2 times, this is 0.68 times 0.32. No, I said it was 44%, didn't I? So, sorry, this is 0.44. So that's going to be 0.90, which I could have also just gotten by subtracting from 1, times 2,000. That's going to be 1,800. So homozygous dominant, I was already figuring that out there. So 0.46 times 2,000 is going to be equal to 920. And then heterozygous is 0.44 times 2,000. That's going to be equal to 880. Add those two together, I should get 1,800. This doesn't match up with that right there. So a little bit roundabout, but I did get there eventually. Okay, for your reflective practice then, here's what I want you to do. It's a series of practice problems, and I will post these all on Google Classroom as well. And then we will go over the answers the next time. So on this one here, what I want you to do is calculate the change between 1985 and 2011. So you really need to calculate the little frequencies there. You can go and do the other ones as well. There is a graph on the next slide, though, that will show you that as well. So, okay, so you've got the hypothesis. So identify the hypothesis the scientists were likely testing here, and then based off this graph, propose an explanation for the trend, and then justify your explanation. In this case, there is actually something called the correlation coefficient calculated for you. That's the line of best fit and how well that line fits the data. So in this case, the correlation coefficient is 0.7866, which actually means there's a pretty strong relationship between these two variables. So anyway, I hope you're doing well, and I look forward to seeing you next time.